Good afternoon, everyone, and welcome to SenzaGen's Medical Device Webinar. This webinar focuses on the development of GuardSkin medical device and how customers can use the assay, showing results from two different case studies. Today's presenter is Dr. Rosemary Jenvert of SenzaGen. She's our medical device project manager and product manager for Guard applications. Rosemary has a PhD in cell biology and has worked with biocompatibility as a study director and biological expert for many years before joining SenzaGen. She is also a Swedish expert in ISO TC 194. Again, welcome everyone, and I will now give the word to Dr. Rosemary Jenvert. Rosemary, please go ahead.
Thank you, Tina, for that very nice introduction. I would also like to welcome you all to this webinar, and I'm excited to see that so many of you have signed up for the webinar today and are interested in our new product, GuardSkin medical device. I will start my presentation with a short introduction about the Guard platform and the available assays, and then continue with the development of GuardSkin medical device and examples of how you can use Guard for skin sensitization testing of your materials and medical device products. For those of you that are interested to know more about the development of the Guard platform and the Guard technology, I can recommend a webinar held in April that was more technical, and you can find the recorded webinar on our webpage. Before I start going into details about Guard, I would like to give you a short background about SenzaGen. SenzaGen was founded in 2010, and we are a spin-out from Lund University. We're today 23 employees, and we are a highly multidisciplinary team with 9 PhDs. Our business model is simple. As a research-oriented company, we will continue to develop new assays, but also develop and extend the application domain of current assays, and then transfer the technology to CRO partners and industrial collaboration partners, who then perform the routine assays for their customers. Our lead product today is Guard, which stands for Genomic Allergen Rapid Detection, and this is a state-of-the-art technology platform for assessment of chemical sensitizers. The platform today consists of 4 assays: Guard Skin, Guard Potency, Guard Air, and Guard Skin medical device. We already today offer testing of industrial chemicals, but also more complex samples as UVCBs and mixtures, and of course, now recently, medical devices and medical grade materials. If you're interested in knowing more about testing and different case studies of how we use Guard, we had a webinar in September where we presented this, and you can find it through our webpage. Since 2017, we are listed on the Nasdaq First North on the stock exchange in Stockholm, and we have established collaboration with partners in Europe, US, China, and Korea in order to distribute and license our technology around the world. For those of you that never heard about Guard before, I will shortly present the Guard platform. And for those of you that are already familiar with Guard, please bear with me. Maybe you learn something new. Guard is not a single assay, but instead a versatile testing platform on which we have based several different applications. Today, I will focus my presentation on GuardSkin medical device and also use GuardSkin as an example of how the technology was developed. So here at the top, you see our first assay on the market, GuardSkin, for identification of skin sensitization hazard of chemicals, followed by our recently launched assay, GuardSkin medical device, which is based on the same prediction signature as GuardSkin and can be used for identification of skin sensitizing potential in polar and non-polar extractions of medical devices. We also have the Guard Potency assay, which can be used for sub-classification of skin sensitizers according to CLP. And last, the Guard Air assay for identification of respiratory sensitization hazard of chemicals. And this assay can be of interest for you that have devices that needs to comply with ISO 18562, and are in contact with breathing gas pathways. In the brackets behind the product names, you can see the numbers of biomarkers that we use in the different assays. I will tell you more about this later using GARD-skin as an example. I just also want to add that the different GARD assays can be used alone, but also in combination, for instance, to distinguish between skin and respiratory sensitizers, or to classify the potency of skin sensitizers as 1A or 1B according to CLP. I will now continue my presentation by giving you a short introduction to the technology and how we use human relevant cells in combination with genomics and machine learning. Before I tell you more about the GARD technology, I would just update you on the response to a foreign substance that leads up to allergic contact dermatitis. First, an initial exposure to the skin sensitizer. The chemical first penetrates the skin and interacts with endogenous proteins to form haptin protein complexes. This complex is then recognized by dendritic cells, which in response to inflammatory mediators migrate to the local lymph nodes where they activate allergen-specific T cells to proliferate. Then upon a secondary exposure, the prime T cells, which now have established an immunological memory, rapidly reacts to induce a state of inflammation. This in turn leads to the disease-associated symptoms of allergic contact dermatitis. And in this cascade of downstream events, the dendritic cells play an initial role as a decision maker. Having shown you the pathway for skin sensitization, we now move back to the GARD platform and how it works, starting with the most important step when building an in vitro model, to choose an appropriate biological system that is capable to reflect the endpoint that you try to predict. We know that both skin and respiratory sensitization involves the activation of an adaptive immune system. So we believe that the dendritic cells are a good biological system for assay development for these endpoints, as they bridge the innate and adaptive immune system. As a biological system in the GARD assay, we use a human dendritic-like cell line called sensor cells. These cell lines react very similar to the response generated in primary dendritic cells when exposed to a skin sensitizer or a sensitizer. Then when you have defined the biological system, you need to find a way to see how these cells react to external stimuli. And the conventional way to do this is to look at one or two different biomarkers. But in the GARD assay, we use a different strategy than traditional methods and use a biomarker signature. This image shows the 200 genes of the biomarker signature for GARD skin and GARD skin medical device, the GARD prediction signature. What you see here in the heat map is the different regulations of the 200 genes and the prediction signature when the cells are exposed to a sensitizer or a non-sensitizer. These genes cover a number of different pathways known to be relevant for the development of skin sensitization. We have genes that are involved in the recognition of foreign substances, immunological self-defense mechanisms, cellular stress responses, and also chemotaxis and communication with other types of cells in the immune system. It is also possible to map the genes in the GARD prediction signature to the adverse outcome pathway of skin sensitization and show that they do not only cover the key event 3 of the dendritic cells, but also several different key events, for instance, the NRF2 pathway in the key event 2 and signaling that leads to T cell activation and forms an integrated system that covers the major events associated with skin sensitization. This is how our researchers have used genomics to build a more mechanistically relevant in vitro model. So how is then the GARD assay performed? We have, of course, developed a standard operating procedure for testing with GARD, and I will now show you this in a simplified view to give you an understanding of how it works. First thing we do is to define the concentration that we use for the test, the GARD input finder, where we perform a dose response testing to define the concentration that induce a relative viability of the cells of 90%. We then use this concentration to do cellular stimulation. We perform a quality control of the cells, and then the RNA is isolated from the cells. Next step is then to measure the gene expression of the genes in the biomarker signature of the relevant endpoint. We use hybridization-based technology called nanostring, which works similar to the microarray, but it enables us to measure only a fixed set of genes. And in the case of the GARD skin medical device assay, the 200 genes in the GARD prediction signature. The raw gene expression data is then uploaded to an online-based application where we have implemented a prediction model based on pattern recognition called support vector machine. The gene expression profile of the unknown sample is compared to the gene expression profile of the reference samples in our database. So based on the expression profile on the unknown sample, it will be classified as a sensitizer or a non-sensitizer. I will use this recent data from the validation study of GARD skin assay to show you the benefits of using human relevant cells with genomics and machine learning. The study was a blinded ring trial performed in accordance with the OECD guideline, and it was performed in 3 different labs: the Burleson Research Technologies in US, Eurofins in Munich, Europe, and also at SenzaGen in Lund. This study showed that the assay is easy to transfer to other labs and demonstrate an accuracy of 94% and have a good correlation between false positives and false negatives. This study was published in May this year, and you can find the reference at the bottom of this slide. So having told you about the advantages of using human relevant cells and genomics together with machine learning using the GARD skin assay as an example, I will now tell you more about the development of GARD skin medical device, which is based on the same signature as GARD skin and adapted to fit for testing of extracts from medical devices. Today, testing for skin sensitization is often done in vivo using the guinea pig maximization test, which is only 70 to 75% accurate compared to human data. And knowing that all medical device classified products needs to be evaluated for skin sensitization, according to ISO 10993 Part 1, we wanted to develop GARD to fit also for skin sensitization testing of medical devices and offer an assay with human relevant cells and higher accuracy to get safer products. So how did we do that? First, we identified the criteria or the requirements of the method. It needed to work with both polar and non-polar extraction vehicles as described in ISO 10993 Part 12. And the vehicles that is often used in the guinea pig maximization test is saline and sesame oil. It also needed to be sensitive enough to detect the low levels of skin sensitizers that leach out from the material or material device into the extract. As expected, the saline worked fine in GARD, and when we then started to investigate if we could use oil in the GARD platform, which is known to be a challenge in cell-based in vitro methods. We found several oils that worked fine in our cell system, but due to our sensitive assay, some of them were classified as skin sensitizers in GARD. This is probably due to impurities in the oil that can cause skin sensitization, but it has also been reported that sesame seeds and sesame oil can be skin sensitizers in sensitive individuals. We have so far found 2 oils that work with GARD, a super refined olive oil and a sesame oil. We also wanted to show that GARD was sensitive enough to detect low levels of skin sensitizers in extracts. So we performed an in-house validation of the GARD skin medical device assay with silicone and polyurethane materials spiked with known sensitizers. So the in-house validation was performed using extraction vehicles saline, super refined olive oil, and sesame oil. We used materials, model materials, silicone and polyurethane spiked with 10% of 5 different known strong, moderate, and weak skin sensitizers. And the materials were produced at RISE, the Research Institute of Sweden. As the controls, we used extraction vehicles as negative controls, and the positive control was the extraction vehicle with PPD, a skin sensitizer chemical. The extraction conditions were 0.2 gram per milliliters of the material that was extracted for 72 hours at 37 degrees. Here are the results from the in-house validation, and they are summarized in the table. This was also presented as a poster at Eurotox in Helsinki in September this year. The spiked materials, the silicone and polyurethane, was spiked with the skin sensitizers to aminophenol, cinnamic aldehyde, propylgalate, and phenyl benzoate. We also tested the silicone and polyurethane without any spike chemicals as material controls. We also added 3 commercially available tubes made of silicone, polyurethane, and PVC to ensure that the assay is not too sensitive. As you can see here, all the spiked materials were classified correctly as skin sensitizers with GARD, and unspiked materials and the commercially available tubes were classified as non-sensitizers. These results show that we can use saline and oil as extraction vehicles in GARD skin medical device, and that GARD can detect skin sensitizers extracted from medical grade materials, often used in medical devices. The results from the unspiked silicone and polyurethane, as well as the commercially available tubes, are all classified as non-sensitizers and gives us a reassurance that we don't get false negative results from the mixture of chemicals leaching out from these materials. Having shown you the result from the in-house validation of the product, GARD Skin Medical Device, I now want to move on to tell you how you can use the GARD Skin Medical Device for assessment of skin sensitizers of your medical device classified products or for your medical grade materials. GARD can be used during product development in the R&D phase for selection of, for instance, materials, coatings, or colors. You can also use it together with chemical characterization and in vitro irritation testing to document changes during your product's life cycle, like change of a material, change of a supplier or production site, or changes to your sterilization procedures. And finally, of course, for assessment of skin sensitization of your final product. I will now continue to show you 2 case studies where we have used GARD for safety assessment of skin sensitization of medical devices. So case study 1 will talk about sterilization procedure. So the problem the customer had, that they had a new batch of the product that caused allergic dermatitis during clinical studies. The method that was used to test this, we compared the old batch with the new batch, and the product was extracted in cell culture media with serum for 72 hours and 37 degrees. And then we used GARD for safety assessment of skin sensitizers. The results show that impurities in the new batch of the product were linked to the adverse effects seen in the clinical studies, and the skin sensitizers in the new batch was probably introduced during gamma sterilization of the product. I will now move on to case study 2 and a comparison of different product materials. The problem the company had was that they didn't want to use unnecessary animal testing when they compared already marketed product with R&D projects. So the method we use now here is the GARD Skin Medical Device with extraction vehicles, saline and super refined olive oil. We extracted the products for 72 hours in 37 degrees, and used GARD for safety assessment of skin sensitizers. As you see here, the results is that the products on the market is compared to the product under development. You can see to the left, you have a negative control and then the oil control. And number 3 is the positive control. And then the saline. And then you can see the different products extracted in oil and saline respectively. You can see here that all the products tested as non-sensitizers, which is very pleasing to the customer of these products. And this is also to show you how you can use this method, use GARD for testing for new materials during product development. I will end my presentation with giving you information of how to test your products or materials. The first thing, of course, is to contact us, and then we can help you to choose the testing strategy and the test you need to answer your questions in close collaboration with you. You will also need to fill in a test substance questionnaire with information about your test sample. And the turnaround time from when we get your sample is 4 to 6 weeks for the GARD assay. You also have to choose the preferred testing site where you want to have the testing done. And today we have 3 different license labs, 1 in Europe, which is Eurofins in Munich, 2 in US, Burleson's Research Technologies and MB Research Laboratories. And we also have a global net of distributors that can help you to organize your testing at our different facilities. So this is my final slide, and this is the first page of our web page, where you can find more information about SenzaGen and the GARD technology. I have also added the contact details to me, Rosemary, and to my colleague in US, Joshua Smith. I hope that you now have more knowledge about the GARD skin medical device and how you can use GARD for skin sensitizing testing of your devices to get a more accurate answer and safer products by the use of human relevant cells in combination with genomics and machine learning. And also that you can use GARD not only for your final product, but also during product development and during product life cycle to ensure product safety. By that, I will thank you very much for your attention and open up for questions.